So I think good evening everyone. So I think we can just start the presentation. I think later on people will join. So I hope that the slides are visible. And if you have any doubts or any query in between, you can just unmute yourself or you can type in the chat section. So we can just explain that out or discuss it out in this case. So good evening everyone. Hope you are all doing well. So today I'm going to start the week 5 section of the topic cell culture technologies for the teaching assistantship that is conducted by NPTEL, IIT Madras. So I will just move on to the first question itself. So yeah, the formula of poly D lysine is, so okay, someone raised the hand. Is there any question?
Sir, can you be please little louder?
Okay. So can you hear me now? Hello, can you hear me now?
Yes, sir.
Yes, okay. So coming on to the question number 1, the formula of poly D lysine is, which might be the answer. You can type it in the chat box or you can just unmute yourself and then tell the answer.
Option 3.
Okay, option 3 is there. Anyone else having any option and all? Any answer? Yeah. So you are correct. The answer to the question is option number 3. That is the actually it's a heteropolymer, right? So sorry, homopolymer, right? So homopolymer of D-lysine. So D-lysine is basically an amino acid. So basically these lysine are conjugated together to form a long polymer. So that's why it has a formula of C6H12 N2O in the open brackets N. So this N represents the linearity of the polymer, right? So how many repeating units it's made of. The N actually represents the number of repeating units, basically. So this poly D lysine is actually a homopolymer of D lysine, which is an amino acid. So basically, if you look into why this poly D lysine is important, do you know? Have you? I think you probably know. Can any one of you tell me what are the two types of extracellular matrix that we are using in cell culture based on where they are obtained from, based on the origin? Like what are the two types of or the classification of the extracellular matrix used? Anyone? Based on the origin, where it is obtained from, where it is made, or where it can be obtained directly from nature itself. What are the two types of classification of a extracellular matrix that we are using in cell culture? Anyone? Okay, so I will just point out the answer. So basically we know that the extracellular matrix are of two types, right? It is classified into two types based on the origin, that is natural as well as artificial, right? So natural, like these natural extracellular matrix are those which are found within the nature itself, basically from different organisms. Like examples include collagen, which is which is used as extracellular matrix, right? Collagen is there, laminin is there, desmin is there, what is a gelatin, which is hydrolyzed form of like collagen itself. These are all natural type of extracellular matrix. While the other one, that is the artificial or the synthetic ones, are basically made in the laboratory itself. So these examples include poly D lysine, poly D ornithine, etc. So basically these are made within the laboratory and they are made into a polymer by different chemical reactions and all, and it depends. And basically it is also a kind used as extracellular matrix, and sometimes it can be better as compared to natural ones also, because it is actually synthesized in the laboratory based on our preference, right? So several different advantages are there basically. So poly D lysine comes under the artificial extracellular matrix. What is it? The artificial extracellular matrix type of the artificial type of extracellular matrix and all. So basically this is the structure of a polylysine. Basically I said that it is made up of a repeating units of D lysine, right? So lysine consists of two different enantiomers. One is the D and L form, and the D lysine is naturally occurring while the L form has to be synthesized within the laboratory. So poly D lysine is a synthetic amino acid chain that is positively charged. Basically the amino group here, right? You can see that the actually the amino acid structure, you know that it consists of a central carbon atom. It consists of a amino group attached to a carbon atom and also a COH group attached to a carbon atom, and there is a hydrogen atom that is attached to a central carbon atom. And then the central carbon atom is preceded by a fourth group which is called as an R chain. The R chain can differ based on the amino acid that you are studying basically. So in this case, lysine consists of this particular chain. There is a CH2 group here, there is a CH2, another CH2 and another CH2 followed by a NH2 group here. And because it is a polymer, it loses this hydrogen to another OH group, and then it forms a linear chain of this D lysine to form what is called as a poly D lysine there. So it is basically positive charge because of the presence of this NH group there. It gets protonated to yield a positive charge. So this positive charge, because of the presence of this positive charge, and as it is made up of a repeating units of amino acid, it is used in cell culture, especially in case of cell attachment, right? Because if you are using a cell culture plate and all, if the cell is a adherent cell type, if it needs to adhere to a extracellular matrix to show its cell attachment as well as proliferation, then this particular type of extracellular matrix is needed. In this case, in case of non-adherent cell type, it is not needed basically. So poly D lysine is kind of a synthetic extracellular matrix that is used in this kind of coating the cell culture plate and all. And it is a homopolymer of amino acid D lysine, and the chemical formula here, you can see the chemical formula here as it is shown. And based on the extent of degree of polymerization, this N actually changes accordingly basically. So this is actually the answer, that is C. The formula for poly D lysine is bracket open C6H12N2O bracket close followed by an N. So does anyone have any doubt? If anyone has any doubt, you can clear it out, no issue. You can type in the chat box, you can unmute yourself and then ask the question, no issue there. Okay, so I think it's quite clear. So I will just move on to the next question then. So coming on to question number 2, the lysine in poly D lysine is which one with the answer? Glucose derivative, amino acid, phospholipid, or the peptide? Which might be the answer?
Amino acid.
Amino acid. Any other option? Yeah, so basically you're right. So answer is amino acids. Basically in the earlier question also, like it was dictated there. So this is a structure of D lysine there. So you know that there is a central carbon atom here attached to a hydrogen group, which is not shown here. So there is a amino group there, and there is a COH group there also, and followed by the R group, which consists of this particular chain that is in case of D lysine basically. And so basically it's an amino acid, right? So it forms a polymer of poly D lysine and all, so which is used as an extracellular matrix there. So anyone has any doubt there? Should I repeat it again, or anyone has any doubt, you can just ask it out. Okay, so I think it's clear. I will just move on to the next question. So question number 3, the linkage between poly D lysine and cell is, which might be the answer? First one is electrostatic, second one is covalent, biological, or no interaction at all. Which might be the answer? The linkage between poly D lysine and the cells is, first one
Electrostatic.
electrostatic. I got the answer. Anyone else? Any other option? Yeah, you are right. So the answer is electrostatic there. Basically, if you look into the poly-D lysine structure, right? So I have earlier pointed out that this is the structure. So basically, the amino group that is present within the poly-D lysine tends to be protonated. So once it gets protonated, what it will occur? It gets acquired a positive charge there, right? So when a positively charged material comes in contact with a negatively charged material, it has a tendency to attract to itself, right? If it comes in contact with a positively charged material, it will get a repel feature. When it comes with a negatively charged material, it tends to attract to each other, right? So poly-D lysine has that innate, because of the protonation tendency of amino group within the poly-D lysine, it has a tendency to interact with negatively charged cells there. Basically, if you look into the structure of a cell, you can see that there are several electrostatic interactions that are present within the cell membrane of a particular cell culture that you are using, basically. So basically this poly-D lysine, because of the presence of this or the protonated form of this poly-lysine, has a tendency to attach to the negatively charged molecules that are present within the cell membrane itself. In most probably the inner leaflet of the plasma membrane has this negatively charged. So these negatively charged and is imparted by the presence of several phospholipids. Negatively charged phospholipids are there. You know that the cell membrane is composed of different lipid molecules, right? So it is both hydrophobic as well as hydrophilic in nature, while the inner leaflet consists of some of these negatively charged phospholipids there. So the phospholipid molecules actually impart an indirect or the negative charge to the cell membrane, which in turn has a tendency to bind to the positive charged poly-D lysine, which is provided as an extracellular matrix within the culture plate there. So because of this presence of interaction, this particular interaction, because as the interaction is taking place between two oppositely charged molecules, right? Two oppositely charged entities there, right? So it is an electrostatic interaction there, not a covalent interaction, not a biological interaction. It's an electrostatic interaction there. So because of the presence of this interaction, the cell actually binds to the extracellular matrix that is provided by this poly-D lysine, and then after this attachment, the cell adhesion is completed. When cell adhesion is completed, the cell starts to proliferate as well as divide, and whatever the function it needs to perform, it will perform because of the presence of this adhesion there. So basically, this is a type of adhesion that is promoted by strong affinity for this negatively charged protein as well as the negatively charged, cell membrane there, the phospholipids there. So the answer to the question is the linkage between positively charged poly-D lysine and the negatively charged cell membrane is electrostatic in nature. So anyone has any doubt? Or this should I explain it once more, or anyone has any doubt, you can just type in the chat box. Okay, so I think it's quite clear, so I will just move on to the next question then. So coming to question number 4, the dash on what on poly-D lysine interacts with what on cells or protein. First one is positive charge, second one is positive charge, negative charge, third one is negative charge or positive charge, last one is negative charge or negative charge, which might be the answer. If you know the previous answer, you will get the answer to this question itself.
Positive and negative.
Okay, I got one answer, positive and negative. Any other option? Okay, option 2 I am getting also same option. Okay, so I'm getting the answer option number 2, that is positive charge and negative charge. So you are basically right. So the positive charge on poly-D lysine interacts with the negative charge of cells or protein. So basically, like if you say why the positive charge on poly-D lysine interacts with the negative charge, so I have earlier cleared this answer out, so I will just repeat it out in this case. So if you look into a plasma membrane, basically it consists of different leaflets, right? One is the inner leaflet, one is the outer leaflet there. So basically, you know that the plasma membrane consists of a polar head as well as a non-polar tail, right? So it is basically both hydrophobic as well as hydrophilic in nature. But the lipids that constitute this plasma membrane is kind of charged in nature. So basically, if you look into the inner leaflet of the plasma membrane, majority of these phospholipids are anionic in nature. So basically, from the term anionic, you know that it is negatively charged, right? So phosphatidylserine, phosphatidylinositol, several like what is it, phospholipids are there, which are basically anionic in nature. While the outer leaflets of the plasma membrane tends to be positively charged in nature, and also due to the presence of a charge potential difference, because there are several and also apart from this, because of the presence of several ions there, right? So like sodium is present, potassium is present, chloride is present there. So basically potassium is usually present inside the cells, right? And sodium and chloride is basically present in the outermost environment. So also the movement of these ions from to and fro into and outside the cell actually also creates a potential difference there. So basically these overall overall movement of ions, as well as the presence of these anionic phospholipids and other negatively charged protein, actually makes the inner leaflet of plasma membrane much more negative in nature. And because of the presence of this negatively charged phospholipid in nature, they have a tendency to adhere to much of the positively charged extracellular matrix proteins that you are providing there. In this case, the poly D lysine there, because once it is protonated, it is positively charged in nature. So this because of the presence of this negatively charged groups there, the plasma membrane or the cell has a tendency to bind to this positively charged extracellular matrix there. In other case of like other synthetic materials or other naturally occurring extracellular matrix, the interaction might be different, but in case of poly D lysine, it is basically because of this allostatic interaction and all. And this in turn facilitates the attachment or the adhesion of this cell into the extracellular matrix. And if it is a adherent cell type, and once it adheres to the extracellular matrix, it starts to divide and proliferate and carries out the whatever the functions it is necessary for it to carry out within the cell culture that we are providing. So basically, because of the negative charge presence of plasma membrane, the poly D lysine, positive charge poly D lysine actually binds to the cell itself, promoting cell adhesion and proliferation. So does anyone have any doubt regarding this? Okay, so I think it's quite clear. I will just move on to the next question then. So coming down to question number 5. Why is poly D lysine preferred over poly L lysine? First option is because poly D lysine is naturally occurring and economical. They are same and either one can be used, and none is preferred over the other. Poly L lysine is naturally occurring and more prone to enzymatic degradation in culture than poly D lysine. Poly L lysine causes immune reaction to a cell, while poly D lysine does not. Which might be the answer? Why do you think?
Option.
Okay, option number 3 I am getting. Any other option? Yeah, so basically you are right. The answer to this question is that poly L lysine is naturally occurring and is more prone to enzymatic degradation in culture than poly D lysine. So basically, if you look into the first option itself, you know that because poly D lysine is naturally occurring and economical. So one point is right, is basically economical as well as cheaper as compared to poly L lysine, but it is not naturally occurring. D lysine is naturally occurring because it is an amino acid there. But in order to produce this poly D lysine, which is a homopolymer of D lysine, several chemical reactions have to be carried out in the laboratory, and then it has to be made there. So it's not naturally occurring, it's actually artificial. That's why it comes under the name artificial extracellular matrix, right? So as compared to poly D lysine, it's economical, but it is not naturally occurring, it is synthetic in nature. And also basically it is cost effective based on the limitations that poly L lysine cannot be like the amino acid. L lysine has to be made from D lysine by several chemical reactions. That is also laboratory made, and then poly L lysine has to be made. So it is not much cheap as compared to poly D lysine. Poly D lysine is much more cost effective as compared to poly L lysine because of this. And also poly D lysine, you see that it is more prone to, poly L lysine is more prone to enzymatic degradation. That is right, basically. So basically, I think because of the chiral nature of this poly D lysine, right? So it is basically chiral molecule. It can exist as D as well as L enantiomers. So the D, because of this D form of like a plain polarized nature of poly D lysine, it can exhibit much greater stability against enzymatic degradation as compared to poly L lysine. And based on the, if you are using a cell culture for a long time, right, if you are keeping a cell culture for a long time, if you want to use that, then you need to have a much more stable molecule which can be used as an extracellular matrix there, right? So without this we cannot have a proper stable extracellular matrix there. So in order to do that, poly D lysine is much more preferable, might be because of the difference in chirality there. Poly L lysine might be attacked by several proteases there, and then it might be cleared out as compared to poly D lysine, where enzymatic degradation is much less in nature. And because of this, poly L lysine is limited to several other like applications and all. And also poly D lysine is less likely to induce immune response and all, because you are using a cell culture there, right? So in order to maintain a proper cell culture there, immune response to the cell might be less in that case if you are using any immunology for that, and for any immunological purpose and all, or any drug delivery applications and all, you need to have a molecule which induces low immune response there. So poly D lysine is less likely to induce much less immune response as compared to poly L lysine. And also you can see that it is composed of, the two molecules are two different enantiomers. One is made up of L lysine, poly L lysine, and poly D lysine is made up of poly D lysine itself, which is naturally occurring in nature. So another form is that D lysine is a plain, both of them rotates the plain polarized light, because of the enantiomeric difference there, poly D lysine, the D lysine molecules actually rotate the plain polarized light clockwise, while L lysine actually rotates it anti-clockwise itself. And these are some of the important points to remember why poly D lysine is used in compared to poly L lysine. So anyone has any doubt? You can ask question if you have any doubt there, because if you have any doubts to or query to clear out, right, it's better to clear it out at this point. You can just type in the chat box, or you can just unmute yourself and then ask the question. No issue in that case. Can be right or wrong, no issue. Okay, so I think it's quite clear. Basically, I will just move on to the next question. So coming on to question number 6, gelatin is a hydrolyzed form of laminin, ornithine, poly D lysine, or collagen. Which might be the answer?
Collagen.
Collagen, right? Option number 4. Any other option? Okay, I'm getting option number 4. So basically you are right. The answer is collagen. So gelatin is basically a hydrolyzed form of collagen. So basically, if you look at a collagen, basically it's a protein. It's a connective tissue that is found, not connective tissue, sorry. It's basically a type of extracellular matrix that is found within the connective tissues of our body, right? So in case of horn of like several animals like cows and all, it is basically found there. And basically, if you look into the composition of the amino acid, basically it's a protein, right? So it is made up of amino acids there. So it is made up of 3 different type of amino acid: proline, glycine, as well as hydroxyproline. And based on the composition of the collagen there, this amino acid composition will also vary there. So it exists as a triple helix, and then this triple helix actually joined together to form that particular collagen structure there, which actually impart the structural integrity of that particular whatever organ that it is present there, so the connective tissue that is present there. So basically, if you hydrolyze collagen, then it gets formed into smaller, shorter peptides, right? Because you are breaking the bonds there. So when it is hydrolyzed there, it is an irreversible nature, and it forms these smaller peptides, and then it joins together to form what is called as a gelatin there. So basically, it is a gelatin uses collagen as its precursor to get formed, right? So basically similar to collagen, it has also has the similar properties because it is, even though it gets hydrolyzed, the same amino acids are present there, right? So basically it is used as a extracellular matrix also there, and it is for hydrolyzed form of collagen there. Gelatin is a hydrolyzed form of collagen there. And based on some other, like if you are combining it with other hydrogel, which basically is synthetic in nature, and then it can also be used as a extracellular matrix there. And yeah, so it is kind of a gelatin is kind of a naturally occurring extracellular matrix there, not a synthetic one. Even though it is a hydrolyzed form of collagen there, it is directly made from collagen and not due to any other laboratory process and all. So basically gelatin is a irreversibly hydrolyzed form of collagen, wherein the hydrolysis reduces the collagen fibrils into smaller peptide. So laminin is also another kind of naturally occurring extracellular matrix there. Ornithine is basically used as a cementing material for laminin to grow on, because laminin itself cannot grow on a glass material. It needs something else like an ornithine present there in a specific composition there, so that it can sit at the top and then it can act as an extracellular matrix there. Poly D lysine we have actually discussed. So yeah, gelatin is a hydrolyzed or a irreversibly hydrolyzed form of collagen. So does anyone has any doubt there? Okay, so I think it's quite clear. Basically the answer is collagen. So coming to question number 7, this is a microscopic question basically. To observe the structure of nanoparticle, one can use, first one is option number 1, phase contrast microscopy, hand lens, electron microscopy, or a confocal microscopy, which might be the answer. Okay, option number 3 I am getting. Any other answer? Yeah, so the answer to this question is option number 3, the electron microscope. So basically you know that the other microscope cannot go up to a magnification of nanoparticles, right, because it has nanometer scale. So the only microscope that can achieve this particular type of magnification is an electron microscope. So basically if you see an electron microscope, like it's a highly sophisticated microscope, right, just if a light microscope or any compound microscope or any kind of confocal microscope that is said here uses light beam to detect or observe the images of a specimen, right? So in this case, electron microscope, from the name itself, it uses a beam of electron or an electron beam to pass through an specimen, and then to achieve a high resolution image based on the scattering of electrons that are taking place in there, producing a visualizing or producing an image which is at an atomic scale itself. So basically because of this feature, because of the what you say, so yeah, so I will just describe this there. So basically this is a structure of an image. You can see this is the basic like instrumentation, how the electron microscope is actually made there, right? What are the components of a microscope there? So basically if you see that there is an electron gun there, basically it does not use any light wave to produce an image. It just use an electron beam there. So this electron gun actually produces electrons in a beam there. So basically common source include thermionic emitters, or basically tungsten filament, or a field emission gun. These are basically the electron source which when undergone through a high voltage it produces a beam, it releases out a beam of electrons there. So basically there is an anode there, right? Basically electrons are negatively charged. Basically in order to concentrate that into a beam we use an anode there basically. And then there's actually the electron microscope is stored within an vacuum environment there. There should be no air that is present there, because if there is any air that is present within the instrumentation compartment of the electron microscopy, chances are that when the electron beam hits a air particle, then it gets scattered. Basically we don't want that. Basically we need a linear path of electron beam there, right? So basically it passes through this electron anode and then into magnetic lens, which actually concentrate and focus the beam onto the specimen itself. So the beam focusing is done by the presence of this electromagnetic lens. In case of normal light microscopy, we use different kinds of set of lens and all, right? So without this, this used by this kind of electromagnetic lenses, which is powered by this electromagnetic anode, and then it focuses this particular electron beam into the specimen itself. And when it focuses this electron beam into specimen, you can see that there are different type of scattering that is happening there. One is a back scattering there, next is the secondary scattering there, and also X-rays are also produced there. So if you look into a first type of scattering, that is back scattering electrons. So these are high energy electrons. These penetrate into a sample so that you can get an idea of the whole structure, that is what the specimen is made up of. Secondary electrons are emitted from the surface because of deflection and all. It doesn't enter into a much of the inside of the specimen. Rather, it provides a surface composition or surface characterization, or more probably if you say what the topography of that particular specimen is. X-rays are also generated from this based on the, if you look at the X-ray and based on the wavelength of the X-ray or the deflection it is produced, you can actually get an idea of what kind of elements it actually belongs to. So based on the presence of these electrons that are scattered because of the presence of specimen and electron beam hitting that particular specimen, and based on the scattering of number of electrons there, images are created. Denser areas scatter more electrons and appearing darker in the final image, while less dense areas allow more electrons to pass through, appearing more brighter, and so the scattering is also affected there. And based on this, the image is produced there. The scattered electrons are dictated by the detectors, and based on these differences, the density of the specimen there or the particular area there, the image is actually produced there. So this is how an electron microscopy actually works there. Basically, if you look into, and also one more principle is that why electron beam is preferred, electron microscopy is preferred to look into a nanometer scale. I think this image is quite visible. There is currently like a little bit blur. Sorry for that. So if you look into wavelength of the visible light, right? So it exists under this nanometer scale, which is much more higher as compared to an electron beam there, while electron beam has much more less wavelength there. Because of the shorter wavelength of this electron beam, it is used as a source there, or the detection source there for electron microscope to look into much more clearer details, or look into a specimen with much more higher magnification as thus produced by other type of microscope, including confocal or any light microscope itself. And basically, if you look into, as it has a lower wavelength, it can produce okay, which type of electron microscope is okay, I will just read one more time. Which type of electron microscope is mostly used? Basically, I think it differs, right? TEM as well as SEM. Basically, if you look into transmission electron microscopy, you can see that if you want to obtain a higher in-depth three-dimensional structure of a protein and all, TEM is preferred. Another form of TEM is called cryo-EM. You know, the cryo electron microscopy to detect the protein structure and all. So TEM is used in that case in order to get the overall three-dimensional structure of that particular whatever you are looking at. SEM or scanning electron microscope only look into a surface topography of that molecule, right? So in case of nano, nano biotechnology or any nanotechnology, or we prefer to use SEM because we need to look at the topography and all. So based on how whatever experiment you are doing, that might differ because the application of TEM as well as SEM is different, right? So based on that, it might differ there. So I think it depends on the experiment that you are doing basically. So I think that answer is clear. Okay, so in case of like visible light, the wavelength is more as compared to an electron beam there. You can see the wavelength difference, right? Like much more higher difference there. That's why, because of the lower wavelength of the electron beam, it can produce much more higher resolution or much more higher magnification to specimen that we are looking into. So that's why electron microscopy is basically used in this kind of experience, like observe the structure of nanoparticles and all. So the answer to this option is electron microscope to observe the structure of nanoparticle, one can use electron microscopy. So anyone has any other doubt? Okay, so I think the answer is quite clear. I will just move on to the next question then. So coming on to question number 8, goniometry used to measure the what property of a material, hydrophobic or hydrophilic nature, chemical bonding signature, lattice structure, or antibody staining, which might be the answer. Okay, so does anyone has any doubt? So I think someone raised the hand there. Yeah, I'm getting option number 1, hydrophilic or hydrophobic nature. Any other option there? So this option was, I think, discussed in the previous week itself. So the answer to this question is hydrophilic or hydrophobic nature. So basically, goniometry is an instrument that is used to measure what is called as a contact angle. So it is also called as, if you extend the term, it is also called as contact angle, goniometry there. It's a process called there. So basically, it is a standard method used to quantify the hydrophobicity or the hydrophilicity of a surface there. So basically, if you are designing a extracellular matrix or a cell culture plate, right, you need to know the composition or the what is the surface characterization of that particular extracellular matrix there. So you need to do a how porous the the extracellular matrix is there. What is the composition, what it is made up of, like whatever, whether there is any contamination there or whether it is pure in nature, what are all the composition that constitute that particular extracellular matrix there. And also you need to know the surface characteristics like hydrophobicity or hydrophilicity there. So basically, how do we quantify this? Basically, if you know that whenever a rain droplet falls onto a leaf surface, right, it actually forms a spear there, right? Because if you have heard about the outer surface of the leaf is made up of waxy material, right? Because this waxy material is hydrophobic in nature, whenever a water droplet or a rain droplet actually falls onto the leaf surface, it actually makes a spear. Rather than gets flattening out, it actually makes a spear there. So the hydrophilic material, that is a rain droplet, actually when it comes in contact with the hydrophobic material, that is the wax there, it actually makes a spear. What happens when that hydrophilic rain droplet actually falls on a hydrophilic surface, like any other hydrophilic surface like glass surface and all, you would know that it gets flattened out, or it more or less gets flattened out, and then it starts to spread itself to a other surrounding area, right? So it is no longer a sphere, but it actually tries to flatten itself out. So basically, this particular instrument, that is the goniometer, utilizes this particular property of water droplets or any other kind of material, hydrophilic material, to dictate whether the surface that we are analyzing is hydrophobic or hydrophilic in nature. So suppose that the water droplet, this is hydrophilic in nature, actually comes in contact with a hydrophobic nature. You have said that as if we consider it as a wax, you know that it forms a spherical spear here, right? You can see that the spear here. So if they compare the contact angle, that if you put a origin point here, and if you compare the contact angle of this droplet makes with the surface, you can see that this angle is much greater than 90. So basically it means that if the contact angle is greater than 90, the surface is hydrophobic in nature. If it is hydrophilic, as I said, the droplet has a tendency to flatten out to the outer environment, right? So in that case, you can see that the angle, if you consider this as the origin, you can see that the contact angle is much lesser than 90 itself. If you compare the 90 degree from here, you can see that the angle is much less than that, right? So if it is much less, the angle is much less than, the contact angle is much less than the 90 degree, it is hydrophilic in nature. This is what the goniometer employs. You can see whether the surface that you are providing is hydrophilic or hydrophobic in nature. And if you look into several extracellular matrix, it is mostly hydrophilic in nature, because the cell tends to the adherent cell, if the cell needs to adhere and proliferate, then the cell needs to have a hydrophilic surface there so that it can adhere as well as proliferate. So it gives a surface characterization of the extracellular matrix that you have developed within the culture plate there. So goniometry is used to calculate the is used to measure the hydrophilic or hydrophobic nature of the material there. And you can see this is the instrument there. There are two cameras. There are lights that are coming out of both sides. You can see, and there is a camera that actually records this particular phenomenon there. You can see that there is a particular dripper there. So either the molecule or the water will be added into this, and then it forms a slowly it forms a drop there. Then it falls onto the surface here. So then based on the characteristic or the hydrophobic or hydrophilic nature of the molecule there or the surface there, it tends to form a drop there, right? Then it is characterized or the image is taken there, and then the contact angle is measured there. You can see from the view that how the contact angle is measured there, the images are captured, and then it is measured out using a software or anything like that. And based on that you can determine whether it is hydrophobic or hydrophilic in nature. So I think this is quite clear. So anyone has any doubt regarding this? If I need to repeat it out, you can say that like you can just ask. I will just repeat it out. No issue there. So if anyone doesn't have any doubt, I will just move on to the next question. Okay, so I think I just move on to the next question. So coming on to the question number 9. Which of the following is not an extracellular matrix protein? First option is laminin, next is fibronectin, collagen or integrin. Which might be the answer? Okay, I am getting option number 4. Any other option? Does anyone has any other option in mind? Okay, so so previous question, yeah, there was a question basically blood is hydrophobic or hydrophilic in nature. I think it depends on what you are growing, right? Basically, if you are looking into a cell, basically it is hydrophilic in nature. Basically, you look into a cell culture and all macrophages and all. There are several proteins that are underlying the what you say the blood as a composition itself, right? So basically it is hydrophilic in nature, but there are also several other components that are presenting within the blood, right? That are swimming within the blood, like oil droplets will be there. So cholesterol is also present within the blood. These are basically hydrophobic and hydrophobic in nature, and these fat droplets are basically present there, right? So based on the composition of the protein, I think it is basically like what you are looking at, which component of the blood you are looking at, that might be different. Some of it might be hydrophobic, some of it might be hydrophilic in nature. So I think if you look into blood as a whole, it is a composition of both hydrophobic as well as hydrophilic materials, because it consists of all these cells and other whatever molecules or any chemical components that are present there. And yeah, I think that might be the answer. And even if you drip blood into water, it easily dissolves. There will also be compounds that might be present in minute quantity that might dissolve at all. So I think if you look cumulatively into a blood, I think it's both hydrophobic as well as hydrophilic in nature because of the changes composition there. Okay, so I think that's clear. So anyone answer this question? Like I think we got an option, right? So option number 4, that is integrin, right? So basically, if you look into integrin, is not an extracellular matrix protein, rather it's a cell surface receptor, right? So if you look into a cell, there are several different proteins that are present on the outer surface of the cell. So some of might be receptors, some of might be proteases and all, whatever it is, like whatever the function of the protein it is. So integrin is basically a cell surface receptor. So laminin, fibronectin, collagen, these are also all kind of extracellular matrix protein, natural kind of extracellular matrix proteins there. So if you look into the structure of integrin, so basically it exists as a dimer there, right? So when in the inactive stage, basically it exists as a dimer there. So whenever it encounters its like substrate, basically the substrate is actually made up of these 3 amino acids, that is which consists of this arginine, glycine and aspartic. So if a protein consists of this particular sequence, that is arginine, glycine as well as aspartic acid, So it is said to have an RGD motif because R represents arginine, G represents glycine, and D represents aspartic acid. So this is basically an RGD motif that is present within these extracellular matrix proteins like laminin and all. And this is recognized by this particular integrin, which is a cell surface that is present at the which is a receptor that is present at the cell surface there. And once it detects this particular extracellular matrix or this particular motif, it then dimerizes to form alpha 2 beta 2 composition, alpha 2 beta composition, or the subunit composition of stoichiometry differs in this case. It gets activated, and when it gets activated, it starts a single translation cascade there. Means that the cell is telling itself that it has attached to whatever extracellular matrix that is provided, and whether it needs to carry out any function like cell division or proliferation, it can then carry out because it is getting attached there, right? So the addition is completed there. So basically integrin is actually a cell surface receptor, not an extra component of an extracellular matrix protein, but it rather binds to this extracellular motif, that is the RGD motif there, which is present within this extracellular matrix proteins. So does anyone have any doubt regarding this? Okay, so I think it's quite clear basically, and the dimerization actually is important in this case of integrin. If someone asks a question, you can answer that basically. Sorry, not dimerization, oligomerization. So coming on to question number 10. Which of the following techniques can help you understand the three-dimensional structural features of a material there? The first one is AFM, SPS, FTIR, or TEM. Which might be the answer? I am getting option number 1. Anyone has any other option or any other answer in mind? Option number 2 is also I am getting. Any other option? Okay, so I think the answer to the question is AFM, or atomic force microscopy. So basically it is used to determine the three-dimensional topographic image of whatever specimen you are analyzing or the material you are analyzing, right? So basically if you look into the working principle of AFM, why it is preferred rather than SPS or FTIR or TEM itself. Basically if you look into SPS, which is X-ray photospectroscopy, photoelectron spectroscopy, you can see that basically it's pass a beam. Like I am not talking about AFM, I will clear the other options out first, then I will move on to AFM, given working principle there, why it is used in three-dimensional structural analysis there. So SPS or X-ray photoelectron spectroscopy, basically it passes an X-ray beam onto a specimen that you are analyzing, and when an X-ray passes through each and every component that is present within that particular area or of the specimen itself, based on the composition itself, the wavelength or the electrons that are released from that particular area of the specimen changes. So if you have a particular atom at a particular area, and if a different atom at a particular area, then the electron that is scattered from that two different area will be different in nature. And that is why XPS is used to determine the chemical composition rather than the three-dimensional structural feature of the material there. So SPS is used to determine the chemical composition, not the three-dimensional structure, because when an X-ray, it's a particular atom. So as compared to a different atom, the electrons that are that are scattered from that particular area will be different there. Meaning the wavelength might differ or the scattering might be different there. So based on that, we will get an idea of chemical composition of that particular material there. FTIR or Fourier transform infrared spectroscopy. So you know that infrared spectroscopy use an infrared as a source there, right, to detect the chemical composition. You know that the infrared spectroscopy is used to determine the chemical composition, or most probably the whatever organic molecule you are used there, right. So it is also similar to that of SPS. It is used to determine the chemical composition of that specimen there. And TEM, transmission electron microscopy, we discussed. It is used to determine the three-dimensional structure of the the overall structure of that particular specimen there, rather than the overall specimen or the surface characteristics of the topography of the material there. So AFM, if you look, this is how the atomic force microscopy usually works there. So basically the AFM actually consists of a tip there. The tip is actually made up of like whatever material, I think, I'm not sure, but I need to recall that. I think gold plated tips are there, and this is then connected to a stand there, and this overall mechanism is called as a cantilever there. So whenever it passes through a sample, you know that the sample consists of different terrains. If you look into topography, the terrain might change at different areas there, right? So some of them might be lower, some of them might be higher, and based on that, if you look into when an AFM tip is scanning through the surface of the specimen, you can see that based on these height differences, the AFM tip actually changes its position there, right? And whenever a laser beam is deflected into a point of the AFM tip, and then it is reflected back, it is detected by photo detector. Due to fluctuation of this cantilever, the laser beam that is deflected and reaching the photo detector, that might change their angle also, or the position also there. So based on this, based on the difference in the fluctuation of the cantilever, the reflected beam might also change the position within the detector there also. And based on this feedback loop that is getting into a detector there, and based on these changes or the fluctuations of the cantilever, actually it determines the topographic nature of the map of the sample surface there, because the change within the feedback loop that is getting there because of the laser fluctuation and all, it determines the topographic map or the image of the sample surface there. And hence, because of this, when you are scanning the whole sample surface there, then after that you can determine the three-dimensional structure of that particular material or the specimen that you are analyzing there. So that's why AFM is essential for determining the three-dimensional surface features of the material rather than the other techniques there. So does anyone has any doubt regarding this? It is also called atomic force microscopy. I think it's quite clear, right? Basically, you know some of the basic techniques that from the previous class you might have attended there. So I think it's quite clear. So I will just move on to the next question. Coming on to question number 11. Poly D lysine consists of how many nitrogen atoms? 1, 2, 3 or 4? Which might be the option?
2.
2. Okay. Any other option? Option number 2 again I am getting. Yeah. So the answer to that question is option number 2. The poly D lysine contains 2 nitrogen atoms. So basically this is the structure of the one of the repeating units of poly D lysine. Basically it's a homopolymer of poly D lysine, right? So you can see that the amino group that is attached to a carbon consists of one nitrogen group there, and the R group that is attached to the carbon there consists of a NH group there, which contributes to a second nitrogen atom there. So poly D lysine consists of two nitrogen atoms there. So I think this question is clear. Anyone has any doubt? Okay, so I think I will move on to the next question. So basically a simple question. So coming on to question number 12. Which of the following is a synthetic extracellular matrix? Option number 1, matrigel, 2, collagen, 3, laminin or polyl or anything. Which might be the answer?
4.
Okay, 4 is the answer. Anyone has any other option?
Polyl or anything.
Polyl or anything. Okay, so the answer is right. The answer to the question is polyl ornithine. Basically, if you look into other substances like matrigel, basically matrigel is something that is a kind of a gelatinous protein mixture that is obtained from mouse sarcoma cell. So sarcoma is a kind of what kind of cell? What is a sarcoma cell? Basically, it's a cancer cell, right? Sarcoma cell and so basically it is obtained from this particular material is obtained from this particular sarcoma cell, and it is not synthetic in nature. Basically, it's a natural in nature because of that. And laminin is also a similar thing. This is a natural protein that forms a basal component, but it forms a key component of the basal lamina in the extracellular matrix there. And it can also be synthetic, but in order to make it synthetic, it has to be laboratory modified, and all. Similarly, material can also be made synthetic unless it is laboratory made there. So poly L-ornithine and collagen, you know, we have discussed before, right? It's basically a triple helix, and it is present within the connective tissues and all. So poly L-ornithine is the one that is like synthetic in nature, and based on the name itself, you know that it's made up of a repeating units of L-ornithine there. And basically this kind of materials are used as a base layer for like other compounds like laminin, which can be spread over it, so that laminin can interact with it and then can interact with the cell adhesion and promote cell adhesion and proliferation itself. So I think this question is quite clear. So anyone has any doubt? Poly L-ornithine, poly D lysine and everything, like they are all kind of artificial extracellular matrix proteins and all. So I'll just I think I will just move on to the next question. So this I have been repeated, I think. Question number 13. Gelatin is a hydrolyzed form of which form? Collagen, elastin, lamin or fibronectin. This has been repeated, this question. Yeah, collagen. Yeah. So the answer to this question is collagen basically. So basically if you look into a collagen, it consists of a triple helix, right? And major components are made up of like proline, hydroxyproline as well as glycine. And it forms a triple helix, and this triple helix joins together form to what is called as a like a whole structural component of that collagen itself. So when you are hydrolyzing itself, this collagen forms actually gets irreversibly hydrolyzed into smaller peptides, and these peptides in turn aggregate to form what is called as a gelatin, right? So basically gelatin is a hydrolyzed form of collagen, and because of the chemical nature of this gelatin, it is also used as a naturally occurring what you say extracellular matrix there for cell adhesion and proliferation. And yeah, difference are there in characteristics of gelatin and collagen. So I think it's better we not get into much deeper account on that. So the answer to this question is collagen. Gelatin is a hydrolyzed irreversible form of collagen. So does anyone have any doubt regarding this? I think we have discussed, that's why I have moved to the next question. Okay, so I think I will just move on to the next question. Coming on to question number 14. So atomic force microscopy or AFM we have discussed. Helps us analyze, first option, chemical constituents in the bulk of material, topography of the surface, concentration of material or electrical simulation of cell. Okay, so I am getting option number 2. Any other option? Anyone has any other option in their mind? Yeah, topography of the surface, right? So basically if you look, basically to determine the three-dimensional structure, that's the topography, right? So there's a cantilever there, there's a tip there at the attached to the cantilever. So whenever the tip actually scans through the entire surface of the specimen there, you know that it actually fluctuates or oscillates up and down. And then there is a laser hitting at a specific point of the cantilever, and then it is reflected back, and it is detected by a detector there, right? So whenever the cantilever moves to the surface will be sometimes different there, and the tip also fluctuates in this case. The cantilever also fluctuates in this case, and when it is fluctuating, the laser might change its direction angle there, and when it is detected by the detector based on the changes in deflection or the scattering of electron or the reflection of the electron there, you can actually determine the whole topography of that particular image there, or more probably what you would say, a three-dimensional structure or a picture of that particular material there, of the surface of that material there. So I think it's quite clear from the previous question that we have discussed. I think I have given the principle there. So anyone has any doubt there? Okay, so I think it's quite clear. So I will just move on to the next question. Coming on to question number 15. So this is one another technique that you have here. X-ray photo electron spectroscopy is used to analyze option number 1, chemical constituents at the surface of the material, topography of surface, chemical constituents in the bulk of the material, or topography of the bulk. Okay, I am getting option number 1. Any other option? There are two. It is used to measure the chemical constituent, but there are two options there, 1 and 3. Chemical constituents at the surface of the material or chemical constituents in the bulk of the material. Why can't be it both? Can be both, right? It used to determine the chemical composition of the material there. Anyone has any answer regarding that? Basically, it's used to determine the chemical composition. Why can't it determine the bulk of the material? Yeah, basically you are right, the chemical constituents of the surface of the material. So basically SPS or X-ray photoelectron spectroscopy is primarily utilized for analyzing the chemical constituents at the surface of the material, not the bulk of the material, because it is actually sensitive. In order to determine the chemical constituent, like infrared spectroscopy, it should be sensitive there. So however, it can only probe up to depth of 2 to 10 nanometer. It cannot go more than that, because the light is X-ray is actually hitting at the surface rather than going inside. It actually hits the surface, and based on the penetration capability of the X-ray that is provided there, it can only probe up to a depth of 2 to 10 nanometer there, and from that up to this particular boundary it can determine the detailed chemical analysis or the chemical composition analysis of elements present there, and also the chemical states in this region. And in case of cell culture, you know that if you are developing a particular culture plate there, right? So if you are providing an extracellular matrix, you need to know the chemical composition of the extracellular matrix. Or if you are getting a cell culture plate there, if you don't know the chemical composition, you can use this particular SPS technique or X-ray photoelectron spectroscopy to determine the chemical composition that is present at the surface of the cell culture plate there, and then you can see whether this particular cell culture plate or the extracellular matrix will be essential for your cell culture incubation or culture itself, or growth itself in this case. So it can reveal how different, and also if you are experimenting with different kinds of extracellular matrix, it can also determine which are the better substrate for cell attachment, growth, as well as differentiation, and other techniques how different treatments or coatings might affect the biocompatibility and functionality of cell culture surface, because the presence of extracellular matrix is actually necessary, right? Without the proper extracellular matrix there, the cell might not divide or proliferate at all, or the phenotype might be different as compared to what we are expecting to get. So basically, these kind of studies are usually carried out in the presence of X-ray photoelectron spectroscopy. So this is actually the working principle of a photoelectron microscopy there. Sorry, X-ray photoelectron spectroscopy there. You can see that a focused beam of X-ray is actually hitting this particular sample. This is a silicon dioxide there. So basically consider that is a sample there. Any other sample can be used. So X-ray is actually getting hit there. When the X-ray is actually getting hit from an atom there, right? So it ejects an electron from that atom. So the electron gets taken off from the surface. Then it is detected by this particular detector. So there is an electron energy analyzer there. These photo emitted electrons are getting detected by this detector, and this analyzer specifically analyzes the electrons that are getting emitted from there. And based on the analysis they have done, they generate a plot there, and from the plot we can actually determine the chemical composition there. So it's basically simple in nature. It's similar to that of spectroscopy or whatever other kind of spectroscopy instrumentation that you are using. Just in this case, it is that it uses an X-ray there, and which actually emits out the electron beam, and this electron beam actually dictates the chemical composition that is present within the sample that you are analyzing there. In this case, you can see that it is made up of silicon there, and our limitation is that it cannot analyze hydrogen or helium atoms there. So it's kind of like one of the limitation of X-ray, X-ray photoelectron spectroscopy there. So this is the working principle. So this anyway, and based on the depth that is analyzing, as I said, 2 to 10 2 to 10 nanometer, it cannot go probe into much more depth higher than that. So basically that's why it's used to detect the what is it, chemical. That why X-ray photo electron spectroscopy is used to analyze the chemical constituents at the surface of the material rather than the bulk of the material. So does anyone have any doubt regarding this? I think it's clear. Okay, so I think it's quite clear. So I will just move on to the next question then. Coming on to question number 15. Contact angle on the surface is indicative of dash and what is measured using what. So first one is hydrophilicity osmometer, second is osmolality or osmometer, hydrophilicity or goniometer, and osmolality or goniometer. Okay, I am getting option number 3 of hydrophilicity or goniometer. Any other option? Yeah, this we have discussed earlier also, contact angle measurement, right? So contact angle measurement is used to indicate not just hydrophilicity but also hydrophobicity. It gives a surface characteristics of that particular specimen there, right? And it is measured by an instrument called a goniometer, and the procedure is called contact angle goniometry there. So basically, like if the surface is much more hydrophilic, if a water droplet is dropped onto a surface, then the water droplet starts to spread out, right? So in case of if it is hydrophobic, because the hydrophilic water droplet, when it falls down into the surface, it actually forms a droplet there rather than getting flattened out. So because of this particular difference in the surface characteristics of the specimen and the presence of or the change in the contact angle, whether it is less than 90 degree for hydrophilic compound specimen there, or whether it is greater than 90 degree for hydrophobic specimen there. So based on that, we can actually determine the contact angle, and then you determine whether the surface is hydrophobic or hydrophilic in nature. And we use an instrument, that goniometer there, just to analyze that. So the contact angle on the surface is indicative of hydrophilicity and is measured using the instrument goniometer. So does anyone have any doubt? Okay. So I think I will just move on to the next question. The answer is hydrophilicity and hydrophobility. So sorry for that. So coming on to question number 17. Concanevalin A is a what? A carbohydrate binding protein, protein binding carbohydrate, protein binding lipid, or a lipid binding carbohydrate. Okay.
Carbohydrate binding protein, sir.
Carbohydrate binding protein. So anyone has any, I think everyone is going with that particular option. So the option answer is you are right, carbohydrate binding protein. So concanevalin A or CONA is actually a lectin, right? So carbohydrate is a carbohydrate binding protein. Lectins are carbohydrate binding proteins. So which is and is originally extracted from the jack bean, Canavelia ensiformis. Basically, the name actually comes from that particular scientific name itself. So it is also called as CONA, abbreviated as CONA. So basically, it's a member of the lectin family, and binds to several like structures that are present within the glycoproteins or glycolipids that are present within the cell surface, right? So as I said, cell surface consists of several different proteins, and these proteins can be attached to carbohydrates. So if the carbohydrate moiety is present within these proteins, then it is called as glycoproteins. And apart from that, other lipid molecules are present also, like glycolipids. And basically, if you look into the internal structure of these glycoproteins, there are several molecules like mannose groups are there, glucosyl groups are there, and based on these differences, like there are different like a protein. Okay. So is it naturally occurring? Yes, concanevalin is basically naturally occurring. You can see that it is extracted from this particular jack bean, right? So basically, as it is extracted from this particularly naturally occurring form, then it is basically a present there, and it's a lectin. Basically, a lectin is one of the compounds that are determined first by like what is it? So anyone has any doubt? I think someone raised the hand. And yeah, so I think it was by mistake. So I think lectin is also there. Lectin is basically naturally occurring in nature, and basically it binds to these monosyl groups or glycosyl groups that are present within these glycoproteins or glycolipid. So I think in the class, if you have looked into YouTube videos that are provided there, you might have heard that there are several different rods or cones, right? Rods and cones are present within our eyes, right? So basically these cannot grow individually on poly D lysine or poly D ornithine, whatever you are providing there. However, if you look into a surface characteristic of that particular rods or cone there, it consists of large number of this glycoprotein there. So this concanevaline A along with the other substrates or any other addition there or any other substance there, it is used as a like cell surface coating there or an extracellular matrix to grow these rods or cone there, because presence of this glycoprotein helps concanevaline A promotes the attachment of concanevaline A to it. So if what if concanevaline A is used as an extracellular matrix there, because rods and cone cannot grow on other extracellular matrix there like poly D lysine, they cannot grow. So this concanevaline A is combined with the other matrices there to form the extracellular matrix there. So once it is plated over the cell culture, if you are pouring a culture containing these particular rods and cones there, it will gladly bind to this extracellular matrix there because of the presence of concanevaline A, because of the presence of large number of glycoproteins that are present on the surface of rods and cones. So it is used as a cell culture coating material there and creating an artificial ECM-like environment there. Because of the presence of glycoprotein, any cell that consists of large amount of glycoprotein, this particular cone can be used as an extracellular matrix there, and it can promote this cell once cell is attached to it, it can then promote its growth as well as proliferation there. And yeah, so I think that is quite clear. So concanevaline A or cone is obtained, which is obtained from jack bean and is naturally occurring in nature, is a carbohydrate binding protein. That's why it's called as a lectin there. That particular term is used to determine that particular like carbohydrate binding property of this particular concanevaline A. So does anyone has any other doubt? Okay. So if you have any doubt, no issue, you can just ask it out or type it out, no issue in that case. So I think I will just move on to the next question there. Coming on to question number 18. Retina contains rods and cone cells. Rods dictate what and cones dictate what part of the light. First one is intensity, second one is color intensity, third one is color, and the last one is intensity color. So I am getting an option intensity and color. Anyone has any other option? Or anyone is okay with that option? Option number 2 I am also getting. Option number 4 I am again getting. Okay, so option number 4. Yeah, I think sometimes it will differ in the case. So the answer to this question is option number 4. Retina contains rods and cone cell. Rod dictates the intensity and cone dictates the color of the light. So basically the rods or cones are photoreceptors, right? So they are basically proteins there. Rods or cones are basically cell types there. They gather all these proteins like rhodopsin, like conopsin, etc. So they are basically photoreceptor cells there, right? So if you look into the structure of the rods, so you can see that it actually determines, as you see, the intensity of the light. So basically it does not determine color, that is the function of the cones there. So it is highly sensitive to light, and in case of low light vision, because it has the tendency to detect low intensity of light, it can also detect that, and that particular phenomenon is called scotopic vision. And this detect the intensity of light even in case of night vision and all. When you are looking at night and all, majority of the light that is gathered at that time is because of the presence of rods, not because of the presence of cones there. While in case of cones, basically it functions best in bright light because it gathers colored light there, right? So this is also called as photopic vision there. And basically there are 3 different kinds of cones to determine different forms of light there. So these are called as S cones, M cones, as well as L cones there. So and it determines the color perception there rather than rods, which determine the intensity of light that is entering the eyes, right? Cones actually determine this color perception there. It determines what color distinguish is different from the other color there. So basically if you look into the structure of the eyes there, you can see that at this particular portion in the magnified image, you can see that there are different type of cells, right? So there is a rod cell there. Based on the name itself, you can see that it's kind of a rod-like structure there. In case of cone, it's actually kind of conical structure there. And you can see that it is differently colored because cones have the tendency to determine three different colors only: red, green, as well as blue. RGB, that background, it can actually detect. And based on the presence of this combination of colors, because of the when the light is entering there, like because of the presence of red, green, and color, there we can create the eye can itself create a color perception there. Based on this, they can actually determine the difference of the light that is entering into the eyes there. So actually, those are the different kinds of like cells, photoreceptor cells there. So retina contains rods as well as cone cell. Rods detect the intensity of the light, and cone detects the color or the color perception of the light. So does anyone has any doubt there, or it is clear? Okay. So I think it's quite clear. I will just move on to the next question then. So coming on to question number 19. Rods or cones are electrically active, sound sensor, pressure sensor, or electrically inactive, which might be the answer.
Electrically active, sir.
Electrically active. Option number 1. Yeah, basically you are right. The answer is electrically active because you know that actually it perceives the light, right? So it cannot directly convert the light into a like image itself. Rather, when it receives the light, several electrochemical changes happen within the or electrochemical or electrical potential changes happen within this rod as well as cone cells there. Based on the changes in electrical potential there, or based on the amount of light that is entering or the color that is entering the cell, the changes will be different in this case. And based on the difference in the cell or the electrochemical potential or electrochemical changes there, the image is created within our brain there, and then it moves on to different neural cells, right? Then it moves and gets into our brain, then the images processes, then the brain process these electrical changes and then creates an image that is coming up in our mind there, right? So based on this presence of electrical changes there, it is rods and cones are said to be electrically active because it should carry out these changes in electrochemical potential difference and everything there. So only so that it could actually create a particular image, whatever it is in your mind, so that you can produce a particular image of what or the light which is gathered from the outside there, right? So rods and cones are electrically active. So anyone has any doubt regarding this? So I think it's quite clear basically from the principle, the electrochemical aspect. So I will just move on to the last question, question number 20. The powdered collagenase should be stored at all time desiccated at plus 20 degrees Celsius, 0 degrees Celsius, minus 4 or minus 20 degrees Celsius.
Minus 20 degrees Celsius, sir.
Option number 4 I am getting, majority of you. Option number 4. Yeah, so the answer to this question is option number 4, minus 20 degrees Celsius. Because collagenase is basically, it differs basically. If the lyophilized form of collagenase, if you look into a vendor details and all, if you just type in powdered collagenase in the Google, you can see a different companies that are providing this particular enzyme there, right? So if you look into the information catalog, they also provide a catalog, the substance information, where it should be stored at, what is the quality, how it is purified, everything they will give. So if you look into the storage information, you can see that actually it exists between 2 to 8 degrees Celsius rather than minus 20 degrees Celsius. So in case of long-term use, I think it's preferable to use minus 20 degrees Celsius, because if you are looking at a normal room temperature, or if you are keeping it at a normal room temperature, because of the presence of other enzymes or any contamination that might be happening within the powdered collagenase there, right? So chances are that within each time, if it is in room temperature and if the enzyme or the contaminant is active, it will cleave or degrade this collagenase out. So next time when you are using it, it might show any other functionality at all. So it's always important, even though it is shown as 2 to 8 degrees Celsius, it's always important if you are using an enzyme, because it is a collagenase, right? It ends with ase, it degrades collagen. So whatever enzyme you are using, it is always preferable that you are storing it at minus 20 degrees Celsius if you are using it for a long time, or if you are not using it for regularly, if you are using it rarely in some experiments and all. So the answer to this question is minus 20 degrees Celsius. The powdered collagenase should be stored at all times desiccated at minus 20 degrees Celsius. You can also store it at 2 to 8 degrees Celsius, no issue there, but it is preferable to store at low temperature minus 20 degrees Celsius. It's preferable not to store it at room temperature, because it will rapidly get degraded over time. Degrades, but the process will get rapid. So I think the question is clear. Anyone has any doubt regarding this? So I think we will end the section with today. 20 questions are completed. So thank you for everyone for attending. Okay, our one question is there. So when we use disposable plastic culture vessels, okay, does it affect the growth of cell culture? Okay, I think I need to check the composition of the cell culture place there. I am not sure that how plastic will affect the cell culture and all, because you are basically growing in extracellular matrix, but the surrounding media is actually like made up of polystyrene and all everything, right? So I think I need to look into that, because specific characteristics, if any experiment, it might affect the phenotype, but I am not sure. So I will just check it out basically. So thank you for that question basically. So anyone has any other doubt regarding this, regarding the today's section that you have taken? Okay, you can ask basically. You can just type in the chat box, or you can unmute yourself and ask the question. Okay, basically if you look into the overall aspect of a cell culture, right? Cell culture actually respires itself. It can absorb oxygen, then release out carbon dioxide. So other than O2, like if you are using O2, like it should be essential also, because higher oxygen amount will lead to free radical formation or all. That might cell lifestyle. But in case of carbon dioxide, what carbon dioxide does is that if the amount of carbon dioxide increases, then it can actually combines with the water present within the cell culture, thereby promoting the formation of carbonic acid there, right? So carbonic acid splits into proton as well as a proton molecule as well as a SiO3 molecule, a SiO3 minus molecule there. So chances are that if the number of protons are getting increased there, the pH will also get fluctuated there. So if a number of proton actually gets increased, the pH will be much more less in this case, rather than going into maintaining it at a optimal condition of 7.2 or 7.4, what might be. So because the carbon dioxide level should be maintained there, because of that, in order to prevent the change in pH fluctuations there, because of the formation of this. Okay, so I will just explain it once more, because carbon dioxide actually, if you look into a level of carbon dioxide, you are growing it in if you are looking into a cell culture incubation, right? So you are basically growing it in a CO2 incubator there. So in this case, you can see that the carbon dioxide that is produced within the culture actually combines with the water there to produce what is called as a carbonic acid there, right? H2CO3. H2O plus carbon dioxide gives out H2CO3. So as the level of carbon dioxide increases, the carbonic acid formation will also get increased in this case. So if the carbonic acid formation increases, then the level of proton that is formed from this carbonic acid will also get increased there. This will change the pH of the culture there. So if you have optimum the pH condition of 7.2, 7.4 where you are initially growing the cell, because of the increase in carbonic acid there, the chances are that the pH will go into an acidic range there. So in this case the pH is getting fluctuated, right? So if there is any change in pH towards the acidic range, then it will affect the cell culture growth there. So we don't probably want that. So that's why carbon dioxide actually plays like carbon dioxide level should be regulated in each case there basically. And it is also basically a byproduct, right? So respiration do happens in this case. Oxygen gets absorbed by a cell, then it gets released by the, then carbon dioxide is getting released there. So that's why carbon dioxide level should be maintained there, and that's why it's important for cell growth also. So I think the doubt is clear. Yeah, I will just type my email ID here, I think. So I think you can just contact me if you have any query related to this. My email ID I have just given here. So I think the doubt is clear also. So okay, so yeah, sure, thank you. So, and does anyone has any doubt? You can ask question in time, no issue in that case. Okay, so I think I will just stop the class at this section itself. So thank you everyone for attending. Thank you, thank you. I hope that you will also join the next class itself. So thank you.